Hi everyone, I'm Dora and this is the video for the higher level part of photosynthesis. So the HL part begins with explaining what the photosystems are. Photosystems are areas of pigment molecules that can generate and emit excited electrons. So as you know, photosystems are located in the thylakoid membranes of chloroplasts. Photosystems have chlorophyll molecules which give the green color to chloroplasts, but it's also important to mention that there are other examples of pigments such as keratin and xanthophyll. So in these protein complexes, the pigment molecules absorb light to cause an electron in one atom to become excited and jump to a higher energy level. A specific amount of energy is required for this to happen, which is given by sunlight. After this, when the excited electron in a pigment molecule drops back down to its original level, the energy emitted is absorbed by an electron in the adjacent pigment molecule, causing it to become excited. And this goes on. This process is called excitation energy transfer. This way, energy is transferred from pigment to pigment until it reaches the reaction center in the core complex. So when they reach the core complex, it reaches a special pair of chlorophyll molecules in the reaction center. These molecules are able to donate pairs of excited electrons to electron acceptors, which in this case is oxygen. So basically, first, light energy is absorbed, which generates excited electrons, and then these electrons are emitted from the photosystem, carrying the energy needed for later stages of photosynthesis. There are two types of photosystems in the chloroplast of a plant, with different functions. So in this process, the photosystem 2 is first stimulated. They're both located in the thylakoid membranes, and the first excited electron is from this photosystem. And then it transfers the electrons to a chain of electron carriers, which then gets the electrons to photosystem 1. Photosystem 1 gives the excited electrons to NADP reductase, which uses the electrons to reduce NADP. Also, when an electron is excited in photosystem 2, the electron is replaced by another electron, which is given to the photosystem through photolysis of water. There are significant advantages in having pigment molecules arranged in the structured area of a photosystem. First, photons of light are scattered. Even in a high-intensity light, one pigment molecule would only intercept a few photons per second. So the photosystem combines over 100 pigment molecules, increasing the number of photons absorbed per second by 2 orders of magnitude. Individual pigment molecules only absorb light in a narrow range of wavelength. The range varies between pigments. For example, chlorophylls do not absorb green wavelengths, but keratin does. So keratin transmits orange wavelengths. Energy can only be transferred if the molecules are in a close and precise orientation. So this structure helps the energy to be transferred. So as I've said before, the missing electron in the photosystem 2 is replaced by an electron coming from the photolysis of water. The photolysis reaction is written here. The water gets broken down into oxygen and 4 hydrogen ions with 4 left electrons. Chemiosmosis is the ATP production using a proton gradient. Through the process, protons travel across the membrane down a concentration gradient, and also through ATP synthase. And by creating a concentration gradient that releases energy, the released energy is used to make ATP from ADP. The ATP produced by ATP synthase is then released into the stroma. And here it provides energy for the synthesis of sugars and other carbon compounds from carbon dioxide in later stages of photosynthesis. So here you can see the process of ATP production through chemiosmosis. First, the photosystem 2 absorbs photons of light, and then the excited electrons go to electron carriers, and also the missing electrons are replaced through photolysis. So here a proton gradient is formed. Also, as we've mentioned, the photosystem 1 reduces NADP with NADP reductase by using excited electrons. Through the proton gradient, ADP is turned into ATP by ATP synthase, and this is called chemiosmosis. So let's talk about the reduction of NADP by photosystem 1. Photosystem 1 has NADP reductase, which uses electrons to reduce NADP into NADPH, which can also be called reduced NADP. Electrons that are used to reduce NADP are replaced by electrons carried by plastocyanin. This is an electron micrograph of a chloroplast. So this is one thylakoid, and a stack of thylakoids is called a granum, and multiple stacks of thylakoids are called grana. We have the thylakoid membranes, ATP synthase, photosystem 2, plastoquinone and B6F complexes, plastocyanin, and photosystem 1. This is a diagram showing the thylakoid. This is the thylakoid membrane, the thylakoid space, and this is the stroma outside of the thylakoid. So we have the photosystem 2, photosystem 1, ATP synthase, and the other complexes. Now this is the Calvin cycle. Now here you can see the Calvin cycle. We have 3 general steps, the first being carbon fixation, then carbon reduction, and the regeneration of ribulose bisphosphate. The first step is carbon fixation. So at first we start the Calvin cycle with a ribulose bisphosphate, and then we add a carbon dioxide molecule to it, and the Ribisco enzyme fixates the carbon and makes the molecule 2-glycerate 3-phosphate, which can also be called G3P. So as we can see here, the Ribisco enzyme fixates the carbon dioxide molecules and creates glycerate 3-phosphates. And then the cycle uses ATP molecules and then turn them into ATP molecules, also reduce NADP that came from the first processes and turn it into NADP. And after these processes, 1-triose phosphate is removed from the cycle to be used in other processes. So we can say that the left triose phosphates are reduced versions of these. And these are the processes that reduce G3P. And then we have the regeneration of ribulose bisphosphate. So as we said, we have 5 triose phosphates that turn into 3 ribulose phosphates. And then through breaking down ADP into an ADP and phosphates, the ribulose bisphosphates are regenerated. And this is the final process of Calvin cycle. Using the products of the Calvin cycle, we can synthesize carbohydrates, amino acids, and other carbon compounds. 6 turns of the Calvin cycle are needed to produce 1 molecule of glucose, because each turn of the cycle contributes to 1 of the fixed carbon atoms in glucose. Because glucose molecules have 6 carbons, each of the carbons are fixed in 1 turn, and we need 6 carbons, so we need 6 turns. So we have the light-dependent and light-independent reactions. The first is the process that takes place in the thylakoid membrane, and the second is the Calvin cycle. As I've said, the light-dependent reactions take place in the thylakoid membrane through these processes, and the light-independent reactions, which is the Calvin cycle, takes place in the stroma. So as you can see here, the thylakoid membranes absorb light, do photolysis, transport the electrons by carriers, do ATP synthesis by chemiosmosis, and reduce NADP. And then the molecules needed go into the stroma, which is here, the place outside. And using the reduced NADP and ATP given from the light-dependent reactions, the Calvin cycle is consisted of carbon fixation, synthesis of triose phosphate and other carbon compounds, and at last, the regeneration of ribulose bisphosphate. Thank you for watching this video. I hope it was helpful.